Copyright disclaimer. While watching a video regarding the ingredients of the mpox shot wherein the uploader had exposed a certain ingredient in it, an inactive ingredient had piqued my curiosity. The
human
serum albumin. What's that abomination? Upon checking the NIH Center for Biotechnology information, it has features that are essential for eukaryotic cell culture. Eukaryotic meaning the cells of a living thing that has a nucleus. The HSA is vital in cell survival by scavenging the dangerous reactive oxygen species, making it an excellent choice to promote cell growth. Additionally, maintains a variety of these eukaryotic cells under in vitro culture environment. Kindly bear with me. This HSA is said to be the protein in plasma of all vertebrates. Protein, now we're moving. Then they mentioned a recombinant HSA. Recombinant meaning by combining genetic material from two different sources. Then they mentioned combining yeast. Utter abomination. Down the article, it mentions Escherichia coli or E. coli. That reminds me of what our biology second year high school teacher used to kind of tell the class. Where did the fly touch? In the E. coli, the poop. In conclusion, they view the HSA to be highly desirable for biotechnological applications. Furthermore, a replacement for traditionally used serum. Common, widely used, they used hybrid recombinant,
Chimera.
Their goal is to use it in both in vivo and in vitro. In vitro meaning in glass outside the organism. In vivo meaning within the living. It seems like they're mocking, saying laboratory animal, lab rats, guinea pigs. Another that's popping is the stem cell application. But before moving into that, let's watch a bit of the HSA. The Chinese research team has used the rice seeds to synthesize plasma, transplanting the HSA to rice seeds, generating protein while they grow the hybrid grain, hybrid rice. It's fascinating that they have concerns regarding these diseases, yet they still declare these
magical
terms that we hear all the time. It's a bit intriguing when stem cell videos came up when my search was about the substance of the HSA. The Rokakadosh led me to the fetal stem cell transplantation, presented by T.P. Mackenzie, professor of surgery, proposing a treatment or refuah that is before birth on the
genetic
level.
What we're focused on is putting in stem cells or other therapeutic agents, just using a needle into the umbilical vein to treat various genetic diseases.
Needle, charagma, umbilical, let's take note of that.
These possibilities exist in this decade because of the convergence of a number of technologies. So right now we can do rapid genetic diagnosis using whole exome or whole genome sequencing. We have a much better understanding of maternal and fetal biology, and our repertoire of being able to treat diseases using the cutting edge methods of gene therapy or gene editing are really exploding.
Gene
editing therapy.
The main reason to treat the fetus before birth as opposed to waiting until after the child is born, there are a couple of reasons actually. The number one reason is that the fetus has a very unique immune system, which I'll talk about in a little bit, where they can be tolerized to new proteins that they see before birth. You can also, of course, protect the brain before the blood-brain barrier forms.
Infuriating that they're meddling with its development, testing, poking, researching, experimenting.
And you can access stem cells during their migration, for example, for blood stem cells, migration from the fetal liver to the bone marrow.
Accessing things in the microscopic level.
But the trafficking also happens the other way, in that there are mother cells in the fetuses. And the very interesting thing is that the fetus actually learns to tolerate these foreign cells and proteins it sees during development. So because it sees maternal cells during development, it just so happens that it learns to tolerate the mother's antigens or proteins on those cells before birth. How the mother and the fetus, with their different genetic backgrounds, can tolerate each other.
Incredible indeed, because that's how Yahuwah had designed it.
Is that the fetus's T cells, which are the main transplant-rejecting fighting cells, when they see a new protein, instead of becoming these effector T cells, the red ones, which they would in you or me, they actually are more predisposed to become these green regulatory T cells that confirm tolerance. And that the fact that the fetus is seeing and learning to tolerate these maternal cells forms one of the backbones of kind of the miracle of pregnancy.
Example of Yahuwah's Nisim.
And the concept that you can do a bone marrow transplant before birth had been tried some decades before.
Decades of doing it on those innocent babies.
So the idea is that it's a two-step process. So you have a fetal transplant that induces tolerance. And then you do a postnatal booster transplant.
A booster. You can't make this stuff up. It says on the top, to improve
Chimerism.
containing at least 2 different sets of DNA. They say you are a
Chimera.
when you absorb your twin cells. But that term really means a monster, formed from different parts of animals, or hybrid
Human.
etymologically roots from the monster from Greek mythology that Annie had shared on a video before.
There are a number of ethical considerations, of course, for non-directive counseling, and patient representation is really critical for planning the therapies.
Brushing off this ethical issue. Furthermore, are they using their stem cells in the stem cell therapy that even the local celebrities in the Philippines are undergoing?
Stem cells are created from leftover embryos that are willingly donated by patients from fertility clinics. Unlike tissue-specific stem cells, embryonic stem cells are pluripotent. This means that they can be grown into any kind of tissue in the body.
Willingly donated?
All of the fetal remains that are in the red biohazard bags are kept in a freezer. And I can tell you that it was a running joke in the industry to call that freezer the nursery. And the sit there for however long until the medical waste company comes and picks them up.
Waste department picking them up?
But what are stem cells?
Stem cells are actually cells that come from the umbilical cord. Little umbilical cord is filled with millions and millions of stem cells.
Donating them? Really?
Years and years ago, there was an ethical issue of where the actual stem cells came from. Today, that issue no longer exists because the FDA has put forth very, very heavy regulated rules.
Regulated rules. What are those rules? Much like the HEK 293, giving that yummy taste, using the taste receptor cells that are actually protein, sequencing with a cDNA, complementary copy DNA, synthetic DNA, combining rat DNA with
Human.
DNA, recombinant DNA, cDNA being used in cloning, combination, hence they call it
Chimera.
recombinant proteins, widely utilized by bio, but that pharma approved, regulated.
Gene.
Therapy again, meddling with the design. Abomination. It's written, if we want to have that Hayei Olam, we need to shamar hamitzvot, guard the commands of Yahushua HaMashiach. For he came in his Abs Sham. Yahuwah Abinu. Kol lakabud b'shmu. Shalom shalom.